everyone, my name is Leen and today me and my partners Tala and Reem will present to you this Pacademia session regarding your second biology quiz. First of all, how was your first quiz? Good? Are you sure? Okay. So this chapter is a long chapter, but don't worry, it's easy. We're gonna ask you some questions, so try and answer to get candy. Okay? What's a cell, guys? What do you think a cell is? What's a cell? Yes, a cell is the smallest unit of life. So when you describe the property of a cell, you describe the property of life. You have something called the cell theory. It's made up of 3 points. The cell is the smallest unit of life. Secondly, all organisms are made from one or more cells, like unicellular organisms or multicellular organisms. What's an example of unicellular organisms? Bacteria. What about multicellular? Humans, animals. Okay. Cells only arise from other cells. Example, mitosis and meiosis. What's the difference? Who can tell us the difference?
Meiosis only for the sperm and ovum.
Yes?
Mitosis for
any type of cell.
Yeah.
Yes. Okay. Move on. You have something called the principle of complementary structure and function, meaning the shape of the cell will determine how this cell will function. For example, you have RBCs, you have macrophages, you have muscle cells. Okay, they function differently because of their shape. Okay, you have 3 cellular compartments. 1, the cytoplasm, 2, the nucleus, and finally the plasma membrane. The cytoplasm is the intracellular fluid inside the cell, inside the organelles. The nucleus is at the center of the cell. It has the genetic material, and the plasma membrane is the outer boundary of the cell. It's what covers the cell from outside. The plasma membrane, we said, it's what covers the cell from outside. It will control what enters the cell and what leaves. You can also call it the cell membrane. Look at the functions. Let's read them together. 1, physical barrier. It will protect it against toxins and bacteria. Second, selective permeability. What does this mean, guys?
Determines what enters and what leaves.
Sorry?
The cell. Determines what enters and leaves the cell.
Yes, correct. So not everything can enter the cell, and not everything leaves. It's selective. It will select what enters and what leaves. Second, it will communicate with other cells, and it will recognize other cells. So this is a simple picture of the plasma membrane. It's a phospholipid bilayer. Let's dig in deeper. You have 2 types of lipid components in the membrane, phospholipid and cholesterol. Phospholipid, can you guys see? Okay. It has 2 parts, the polar hydrophilic head and the non-polar hydrophobic tail. What does the word polar hydrophilic mean?
Like water.
Sorry?
Like water.
Likes water. What about non-polar hydrophobic? Hates water. So, the head that loves water or water attracting will face outward or inward towards the extracellular fluid or the intracellular fluid. While the non-polar hydrophobic fatty acid tail, it will hide in between. Please go back. Look here. The head loves water, faces outside or inside the extracellular fluid or the intracellular fluid. And the fatty acid tail, they will hide in between. Why do we have this arrangement? Why do we have this arrangement, guys?
To keep the membranes separated.
Yes, to keep the membrane separated and it prevents water from entering. Second, you have cholesterol, also has a polar and non-polar region. The polar region is the hydroxyl group, and the non-polar region, it's basically the ring-fused system. You have 3 functions, please know them. The cholesterol will stiffen the membrane, maintains fluids, and decrease water solubility, not increase, decrease water solubility. Now, protein components of the membrane, you have 2 types, integral and peripheral. What does the word peripheral and integral mean?
Integral, embedded, peripheral.
On the side. So integral or in, it's embedded firmly into the membrane, peripheral on the side. So it's not embedded. Okay. Integral proteins, you have 2 types. Either it protrudes from one side of the membrane or protrudes from both sides of the membrane. If it protrudes from both sides of the membrane, you call this transmembrane protein. Okay? It will span the entire membrane. While peripheral proteins, they're not embedded, so they're anchored to the membrane, either by an anchor or attached to the integral proteins. Let's look at the functions. Integral proteins, they function as channel or carrier. What does this mean? What's a channel?
Pathway.
pathway. It will allow something to move either from inside to outside or outside to inside. تمام? So you need a transmembrane protein for this. Can you have peripheral proteins act as a channel or carrier? Why?
Because they are not embedded in the
Yes, exactly. They're not embedded into the membrane. تمام. You can also have enzyme receptor involved in signal transduction. What signal transduction? You're not giving out candy. What signal transduction? What signal transduction?
the process where cell receive the process and transmit signals.
Yes,
The transmission of molecular signals from cell exterior to cell interior.
correct. Like hormones. Okay. Please, Jawad, give her a stamp. Okay. Peripheral proteins, they function as, they act as enzymes or motor proteins and they link cells together. Guys, can you see this image? Is it clear? Okay. So this one, it's an integral protein. It will protrude from both sides. This is a transmembrane protein. This one is also an integral protein, but from one side. The other one, this is attached outside. This is not embedded. This is a peripheral protein, and this is a peripheral protein. Okay, you have this image in your book. Please read it. You have different protein functions. We said transport, like channel or carriers, enzymatic activity, receptor for signal transduction, like hormones, for example, and attachment to the cytoskeleton and extracellular matrix. This is because it helps in cell movement or the cell shape, the cell shape. So this is very important. Membrane carbohydrates and the glycocalyx. What's the glycocalyx? Let's ask from here. What's the glycocalyx? Anyone? What's the glycocalyx?
Carbohydrates that touch the outer membrane and they act as identifiers.
Correct. So the glycocalyx is a sticky carbohydrate layer, also called a sugar coating. Basically, it lies on the outside and it acts as an identity molecule. Let's say I have a cell. The cell, let's imagine the cell as a person. This person, he's wearing a red shirt or a green shirt. So I remember or I identify this person by his shirt. Oh, he's wearing a green shirt. So it acts as an identity molecule. Important note, it's found on the extracellular surface, not the intracellular. Please remember this and keep it in mind. Do you guys know what's a cancer cell? What's cancer? In the back, what's cancer? Yeah, what's cancer?
Sounds like we think about it.
Exactly. So have you asked yourself, why doesn't our immune system just kill those cancer cells? This is why, because the cancer cell glycocalyx, it will continuously change and change to avoid destruction from the immune cell. So this immune cell, it's not going to recognize the cancer cell because it's changing its glycocalyx. That's why cancer is so dangerous, guys. Okay, go on. This picture in your book summarizes everything I've just said. We said lipids has two parts, phospholipids and cholesterol. Phospholipids, the polar or head region, loves water. Non-polar, tail, hates water. Cholesterol stiffens the membrane, decreases water solubility. Protein, many function like transport. Carbohydrates, as he said, the glycocalyx or sugar coating acts as an identity molecule. Okay, now, do you have any questions? Okay, I'll ask you questions. If a human cell were to increase the amount of cholesterol embedded within its plasma membrane, which of the following would be most likely to happen? Is it A, the plasma membrane would become more fluid and the phospholipid less stable? B, the plasma membrane would become more permeable to ions, less permeable to lipids? C, the plasma membrane would become more stable, less fluid, and less permeable? Or is it D, the cell would form plaque that would potentially block a blood vessel? Raise your hands, please. What's your name? Yeah. What's your name? Yeah. No, at the back. Yeah. What's your name? Chan. Okay. What do you think the answer is?
C.
Why?
Because it will more stiffen the plasma.
Exactly. Clap for him, please. Okay, guys. This part it's so important. So please try and focus. I tried to summarize it in the slide so you can take pictures of it, but we're gonna send the slides later. Okay, cell junctions. They are structures that connect adjacent cells together, either for communicative reasons, mechanical reasons, or just act as a barrier. Tamam? Look at this picture. You can see the adjacent cells together. They are linked. Okay, next. As I said, this part is important. So first of all, please try to memorize the location it's found in and memorize the other name for it. Okay, like it has two names. Like tight junctions, they're called impermeable junctions as well. Memorize the name and memorize the location. Okay, so tight junctions, they act as a seal or a glue to fuse adjacent cells together to prevent leakage. They are found in the digestive tract and the intestine. حاولوا تفهموا the function to know where its location is. What happens in the digestive tract? What happens in the digestive tract? We digest and we absorb these nutrients. So in order to absorb these nutrients, لازم يكون عندنا seal or a glue to prevent these nutrients from leaking. That's why it's found in the digestive tract or in the intestine. Okay, move on. Desmosomes or anchoring junctions, they act as binding bodies and they are found in areas where you have a lot of mechanical stress, like the skin. In my skin, I have desmosomes to prevent tearing, because the skin is an area of high mechanical stress. Also, the heart. The heart will pump blood to the entire body. It has a lot of mechanical, physical stress and resistance. So I need these desmosomes to prevent these tissues from tearing. Tamam? Move on. Gap junctions or communicative junctions. They transport impulses through specific proteins. Do you know what these proteins are called?
Transmembrane proteins.
No, the name of the protein. See? Connexons. Yes, so they transport impulses for a communicative reason through these proteins that are called connexons. They are found in electrically excitable tissues like embryonic tissue, the smooth muscle, and the heart as well. Question. Who can read? Who can read, guys? Chan, yes, read.
The myocardium, cardiac muscle tissue, undergoes a significant amount of physical stress due to its contraction. You would expect to see relatively large numbers of which of the following embedded
Next
within
question.
their plasma membrane.
What's the answer?
Desmosomes.
Correct. But why not A? What's the function of tight junctions? Prevent leakage. What's the function of gap junctions?
Transporting water.
Exactly. Okay, moving on. Who wants to answer? What's your name? Okay. You're answering the next question. طيب, passive transport. What's passive transport? Another one, غيرها.
From high to low concentration.
Sorry? Yes, from high to low concentration without using ATP. Okay? You have three types: simple diffusion, facilitated diffusion, and osmosis. Simple diffusion is the movement it's not assisted, non-assisted diffusion. While facilitated, it's going to be assisted. Why? Because you're going to use carriers like proteins, channels, or carriers. Osmosis is the movement of water. Okay, please, important note: know which molecules can be transported using which type of diffusion. Know the molecules, memorize them by heart. Here it's a simple picture: simple diffusion, unassisted, facilitated, assisted using proteins. Okay, simple diffusion, we said unassisted movement. It depends on two things: either lipid solubility or the size. Lipid solubility means this molecule is non-polar. Okay, so if it's more lipid soluble, it's gonna transport more easily. The smaller the size, it's gonna transport more easily. So either lipid solubility or smaller size. Here I said memorize the examples. You have gases, O2, CO2, steroid hormones, and fatty acids. Important note, please keep this in mind. I'm gonna explain this at the end. Simple diffusion, it's not saturated nor specific. Okay, keep it in mind. Now facilitated diffusion, we said we have two types: carrier-mediated facilitated diffusion and channel-mediated facilitated diffusion. Carrier-mediated uses carriers. Channel-mediated uses channels. Important note, please keep this in mind. Facilitated diffusion is both saturated and specific. طيب, carrier-mediated. We said simple diffusion will transport what? What will it transport? Lipid-soluble molecules and smaller sized molecules. طيب, what about the other molecules? What will happen? No. This one, carrier-mediated. It's going to transport the opposite. So, molecules that are too large and molecules that are polar or lipid-insoluble. So, lipid-insoluble or polar and too large molecules, يعني substances that are too large, مثل glucose and amino acids. Next. channel mediated uses channels. Channels are transmembrane proteins. Are they integral proteins or peripheral proteins? Integral proteins. It's selective based on two things: the pore size, like this is a channel, this is the pore. Okay, so it's selective on the size of this pore. Also, this pore has amino acids. Did you take amino acids in biochemistry or not yet?
Yes.
Okay, so you know amino acids, they have charges. So it will depend on the charges of the amino acid on this pore and the pore size. They transport ions, and you have two types: leakage channels and gated channels. Leakage channel, it's leaky. It's always open. It's always open and allow free movement. Gated channels, they are not always open. They are controlled. So controlled based on two things: either electrical signals or chemical signals. Again, leakage channels always open. Gated channels controlled, open or closed. Next. Yeah. Okay, answer. What's the effect on the rate of facilitated diffusion through the carrier-mediated transport when ATP is added to it?
C.
Yes.
C.
What's your name?
Hassan.
Hassan. What's the answer?
C.
Why?
Because it's diffusion, it's not the active process.
It doesn't use ATP. It's passive diffusion. When oxygen is delivered by the lungs to the blood and subsequently consumed during aerobic cellular respiration in body cells, what happens? Yes? Sorry? D? You said D? Okay. Yes, this is correct. But what type of diffusion is it? Osmosis. What's osmosis, guys? Water movement or movement of water. It's basically the diffusion of a solvent like water through a selectively permeable membrane. Tamam. It will transport using types of proteins called aquaporins. Aquaporins are transmembrane proteins that are water specific. They will transport water freely and reversibly. Reversibly يعني جوا وبرا وبرا وجوا. Okay? They will transport in a single file diffusion. What does this mean? What does this mean? حبة. Okay, what is osmolarity?
The
Total amount of solutes in a solution. Correct. طيب, I'm gonna show you this picture, this node, then I'll go back. طيب. Imagine I have two solutions. This solution has a higher water concentration and less amount of solute. So this solution, it's more dilute. While this solution, feel less water concentration and higher amount of solute, which makes this more concentrated. Come on? Now, the movement is going to move from a higher water concentration to a lower water concentration. Or I can say it's going to move from a lower solute concentration to a higher solute concentration. It's the same thing. So when I say higher water concentration, I mean I have low solute concentration. Or when I say I have lower water concentration, I mean I have high solute concentration. So it's going to move from low solute concentration to high solute concentration. طلعتوا معي? Go back to the picture. So in this picture, this membrane will allow both water and solute to move. So they're going to move until they reach equilibrium. While here, you have a membrane, it's permeable only to water. Solutes can't move through it. So what's going to happen? Let's count here. Here you have how many solutes? 1, 2, 3, 4, 5. Here you have, let's say 10. Okay? So here you have low solute concentration, and here you have high solute concentration. So it's going to move from low solute concentration to high solute concentration. Here you can see the water moving. Tamam? Okay. Question. Who can read? Yes, read.
Solution A contains 5 grams of sugar per liter, while solution B contains 2 grams of sugar per liter. The solutions are separated by a selectively permeable membrane. If the solvent in both solutions in water, which of the following statements best describes the direction most of the water molecules will move?
What's the answer?
To move from
Try again.
move from B to A.
Let's read it again. We said it will move from lower solid concentration to a higher solid concentration. So you have 5 grams of sugar. This is high or low? High. 2 grams of sugar. This is high or low?
Low.
So from 2 to 5, what's the answer?
B to A.
B to A. Okay, so osmosis from solution B to A because it's lower solid concentration to a higher solid concentration. Next. Tonicity. What's tonicity, guys?
The ability of solution to change the cell shape.
Yes. So, how a solution is going to affect the volume of a cell by comparing the amount of non-penetrating solutes outside and inside the cell. You have 3 types. Isotonic, hypertonic, hypotonic. What does the word iso mean? Equal or same. So isotonic, you're going to have an equal amount of non-penetrating solutes on both sides of the membrane. They are equal. The cell is not going to change. It's going to stay the same. Example, extracellular fluids. Okay, hyper. What does the word hyper mean? High. High of what? Higher amount of non-penetrating solutes outside the cell. What will happen then? Water is going to move from inside the cell to the outside of the cell. The cell will shrink or crenates. Crenates يعني shrink. Okay? Finally, hypotonic. Hypo means low, so lower amount of non-penetrating solutes outside the cell. What's going to happen? Water is going to move from outside to inside. The cell will swell or lyse. Lyse يعني explode. انتوا احفظوها هيك. Hypo. Po يعني pop. So the cell is going to explode. Okay? Again, iso, nothing's going to change. Hyper, the cell is going to shrink or crenate. Hypo is going to pop or swell. Who's reading? Yalla in the back. Girls. Girls in the back. Yalla, yalla. It's okay, guys. You can read. Read. What about? Okay, read. Tala Sahrij. Yeah.
Suppose that the woman
Yeah.
Suppose that the woman ran along distance race in the summer. During the race, she lost a large amount of hyposmotic sweat, more water than salt. You would expect your cells to shrink.
طيب, yes. طب why not D? Why not D?
Why would it sweat?
Exactly. So it's losing more water. So you're gonna have a higher concentration of solute. So this cell is gonna shrink. مو بس تقرأوا يلا hypo خلاص it's gonna swell. No, read the question, guys. Okay? Next. Guys, this part it's Okay, go back to the question. Here they're not saying you have a hypotonic solution. They're saying you lost hypoosmotic sweat. This means you are losing water. Since you are losing water, we said if you have a lower water concentration, you're going to have more solutes. So you're losing water, you're going to have a higher amount of solute outside the cell. What type of this? This is hypertonic. Hypertonic. Because it's what?
Why it won't swell after this?
But that's not the thing.
It's not.
No, this is gonna shrink. كيف يعني swell? What do you mean?
Why it's not B? Why the answer is not B? Shrink than swell. Please ask me.
Because now you have a hypertonic solution. Okay? You ready to one? Oh, okay, next. So, as I said, this part is very, very, very important and it can be very confusing. I know this is a single paragraph in your book, but please read it and understand it. I'm going to explain those terms, then we're going to have a short role play, okay? So, you have something called specific. Specific means each carrier will bind to a certain molecule or a specific molecule, okay? Like glucose, for example. So, saturable will mean the limited number of carriers or proteins, okay? You have a limited number of proteins on the outside of the cell. When all of them are busy, the transport will maxes out. Sit down, please, okay? Now we're going to have a little short play. Yes. Where are my volunteers? Let's imagine these 4 guys as proteins. تمام. I'm gonna give each one of them a molecule to transport. Okay. تمام. All of them are transporting a molecule right now, and they are all saturated. They are all busy, and the transport has maxed out. خلاص, this is as far as it can get. تمام. طيب. Now, what's your name? طيب. Hamza will hold a specific molecule. Let's imagine this molecule is a gummy bear, and the rest of them are holding M&Ms. He is specific to this molecule specially. This is called specificity. Okay, again, all of them are transporting a molecule right now. All of them are busy. This is saturation. Okay. Now, if I want to reduce the rate of transport, what will I do? I'm going to remove one protein. Please put aside. Yes, thank you. I'll remove one protein. Now I have less amount of proteins. Okay? So I reduce the rate of transport. طيب, I want to increase the rate of transport again. شو بسوي? Can I, sorry?
More protein.
Can I increase the amount of molecules? Will this help?
No.
Why?
Because saturated.
Okay, even if I increase the amount of molecules, لسه بدهم protein to get transported, صح? So instead I will tell this protein, please come back, transport a molecule, and now I've increased the rate of transport. Okay? Thank you guys. سقولهم. طيب. Simple diffusion. They are not specific nor saturable. ليش؟
Because no proteins are involved.
Simple diffusion.
No proteins.
Yes, no proteins are involved, so they are not saturable. We said saturable means proteins. ما في proteins, it's not saturable. Not specific as well. If a molecule can pass the lipid bilayer, it's gonna pass. So it's not specific nor saturable. Now, facilitated diffusion, it's both. It's both specific and saturable. Why? Because it has proteins.
But leakage is unsaturated.
Sorry?
Leakage channels are unsaturated.
Don't think of it this way. Memorize it like this. It's too complicated to think of it this way. Huh? It's specific. It's specific to the pore size and the charges of the amino acids.
Unsaturated.
Don't think of it this way. Follow the book. Guys, حتى لو في شيء غلط بالكتاب وشات جي بي تي حكى لكم غلط, follow the book. Okay? So we said simple diffusion, it's neither. Facilitated, it's both. Question. Okay. A channel used to take in potassium. The scientists doubled the amount of potassium, but notice that the other side still has the same rate of entry. What rule does this channel follow that limits the rate of entry? Yes? Saturability. Why? Yes, it's transport خلاص صار maxes out and it can't increase further. Which of the following factors reduces the rate of transport during carrier-mediated transport? Increased specificity of the transport proteins, increased concentration difference across the transport proteins, increased saturation of the transport proteins, reduced competition at the transport proteins. By raise of hands, please. Yes? No. No. Okay guys, let's read it. Increased specificity of the transport proteins. طيب عادي. Okay. It will somewhat affect the rate, but يعني to a higher rate. Here we said reduces the rate. So try again. Who can try again?
Wait, I don't know.
Yeah.
You said reduces the rate. When you make it more specific to something, it cannot take the other stuff. So it is technically.
I understand, but here in foundation year you need to read all the options and choose the more correct answer. يعني ممكن في احتمال تلاقوا جوابين they make sense, but choose the more correct answer. Let someone else answer. Yes. Why C? Yeah.
If it's more saturated, it means less number can pass.
If it's more saturated, the transport is gonna maxes out. وخلص, it cannot increase any further. So in this case, they count it as reducing the rate of transport, and C is correct. But let's read this option. Reduces the competition and transport proteins. Does this increase or decrease? Increase. Come on. Yes. Next. Okay, now my partner is going to carry out the rest of the session. Thank you guys. let's start with active transport. So Leen went through passive transport, and we said my colleague Leen went through passive transport, and we said passive transport does not require ATP. So what would active transport require? Okay. So active transport is a process that moves substances into the plasma membrane by using ATP. And usually the substances that move in passive transport were not able to pass through passive transport. So we have three reasons why. First, they could be too large to pass through the channels. Second, they could be incapable of dissolving in the lipid bilayer. So that makes them water soluble, not lipid soluble. And then they could be moving against the concentration gradient. So we said the concentration gradient is always moving from an area of higher concentration to an area of lower concentration. So if we say it's moving against it, it's going from low to high. Now we have two major means of active membrane transport. We have the active transport and the vesicular transport. So let's start off with active transport. So like Leen said, carrier-mediated facilitated diffusion will use transport proteins. So it's the same thing here for active transport, except the only difference between them is that facilitated diffusion always follows the concentration gradient, while active transport will go against the concentration gradient. Okay, so we have an example of primary active transport here, which is the sodium-potassium pump. So let's first look at this picture. Where do we have a higher concentration of sodium? Okay. So we said active transport goes from which to which? Low to high. So it's going to go, the sodium ions will go from the cytoplasm to the extracellular fluid. So the first step here, 3 sodium ions will bind to the pump, and then ATP will be bound. So when ATP is bound, it's going to get hydrolyzed, and we're going to use one of the phosphate groups in order to cause a conformational change, which will open up the pump to the outside, and the sodium will leave. Now, after sodium is released, the same thing is going to happen to potassium. So when we look here, where do we see a higher concentration of potassium? Okay, so we said we're going from low to high. So potassium from the extracellular fluid is going to bind to the pump, and it will open inwards into the cytoplasm, and potassium is going to enter the cell. And then the cycle happens all over again. Okay. Which of the following is the correct role of sodium-potassium, also called the electrogenic pump in a cell? Okay, Reem.
Which of the following is the correct role of the sodium-potassium pump, also called the electrogenic pump? It maintains the concentration gradient, resulting in the rest of the membrane.
Okay, can someone read?
Which of the following is not true about the sodium-potassium pump located in the plasma membrane?
Okay, and if you want to correct this sentence, what would you change it to? يعني هاي الجملة أنت بدك تغيرها تصير صح، شو بتغير؟ صح. Okay, so now that we've talked about primary, let's move on to secondary transport. So we said 3 sodium ions will move out of the cell and 2 potassium ions into the cell, right? And we said that this process will use ATP. So it's called primary because it's the direct user of ATP. When we look at secondary, it's not using ATP directly, but it's using the ATP that was stored in the channel from primary. So in secondary, naturally because sodium has left the cell, so it would want to come back in. So in secondary, sodium will go back into the cell, but it's going to go along the concentration gradient, which we said high to low. But along with sodium, glucose is going to enter as well. So glucose is going to go from an area of lower concentration area, higher concentration. So that's against the concentration gradient. So once they move in together, this is what we call symport. So symport is when two substances move into the cell together or out of the cell, as long as they're in the same direction. And does anyone know if it was in opposite directions, what would it be called? What type of cellular transport system involves the entry of sodium and calcium ions into the same ions in the same direction? No, just answer your question. Okay, that's right. Now that we've went through active transport, let's go through vesicular transport. So we said both of them require ATP, right? So now for vesicular transport, let's just break it down first. So vesicles are basically bubble-like sacs that will either enter or leave the cell. And we also have two other types, which are transcytosis and vesicular trafficking. So for transcytosis, let's say you're going to another country, right? You're going to enter the airport, move through the gates, and then you're going to leave by the airplane, right? So you're moving in, across, and out. This is exactly how the vesicle is going to be moving throughout the cell. It's going to enter, move across it, and then leave from the other side. While vesicular trafficking, you're in the airport already, and you're just moving from one gate to another. So you're not entering nor leaving. So this is how the vesicle will be moving. It will go through different areas of the cell or different organelles. And as we said, they use ATP or sometimes they use GTP, which is just another type of energy with different nucleotides. Okay, so let's start first with endocytosis. So endocytosis, as we said, how is it moving into the cell? In or out? Okay. So we have three types of endocytosis. We have phagocytosis, pinocytosis, and receptor-mediated endocytosis. They differ in the type, the amount, and the method of uptake. Okay, so let's go through the events first. So the substances will pile up at the plasma membrane. So what will happen basically is that the plasma membrane will fold inwards, and the substances that are going to be transported in will pile up inside an invagination of the plasma membrane. Now the plasma membrane is covered by proteins, right? So these proteins are going to surround the invagination, and the substances will keep pushing through until it detaches from the plasma membrane and forms a vesicle inside. Once it detaches, the protein coat will then be recycled back to the plasma membrane, and the uncoated endocytotic vesicle will bind with an endosome. So the endosome is the post office of the cell. It will determine either the material will be fused with a lysosome for digestion, or it will leave the cell. So if it just enters across and leaves, what do we call this? Okay. Now let's start with the first type of endocytosis, which is phagocytosis. So think of phagocytosis as cell eating. The cell is eating large particles. So what happens first is that the substance will come and it will form an invagination, just like we said, of the plasma membrane. And this will be called a pseudopod, or the pod's foot. And then once it's inside the cell, it will then be called a phagosome. So then this phagosome will bind with the lysosome and the substances will be digested. Okay, now for pinocytosis. So pinocytosis is cell drinking. So the cell basically is taking a part of the extracellular fluid. So think of it as if you're drinking a mystery smoothie and you don't know what it contains. So it's basically just taking a drop of the extracellular fluid and it has no idea what this extracellular fluid contains. And lastly, for receptor-mediated endocytosis, so the plasma membrane will have specific receptors for the substances that it will bind to, and no other thing can bind unless it was specific for that receptor. So once it does, the same process will happen again. It will bind with the lysosome and so on. Okay. non-selectively engulfing samples of extracellular fluid, for example, to absorb nutrients, it will likely utilize first one. Okay, and yeah, that's right. And I want you guys to pay attention to something. Here it says non-selectively. So whenever we say non-specific, that means we're talking about phagocytosis and pinocytosis. But whenever they say selectively or specific, what would it be? Now for the events of exocytosis. Okay. So guys, exocytosis is out of the cell. Okay. So Hamza here is going to be the, we have two docking proteins at first. We have the vesicle snare and we have the plasma membrane snare. So what happens is that the vesicle snare will bind to the T snare, or they will fuse together. Okay. Once they do, this will open up. This will open up. This will open up a pore that will let out the molecules inside the vesicle out. Can someone read this question? Guys, I want different hands. Yeah. Okay. See? حدد. 1 and 4. Yeah, that's right. So what does 2 and 3 what are 2 and 3 for? Okay, that's right. Okay. Now for selective permeability. So the plasma membrane is selectively permeable. So what did Leen say about this? What is selectively permeable mean?
Specific.
Okay. So that means that there are some molecules or ions that can leave and some cannot. So whenever a molecule is positively charged, it would be easier for it to leave than for negative ions to enter. Okay? So this is what creates the membrane potential, which ranges from -50 to -90, depending on the cell. And this membrane potential is only at the membrane surface. Please note this, it's not around the whole cell. So once we look at the cytoplasm and the extracellular fluids, they will remain neutral. And the negative charge is only at the membrane surface. Okay. Now for potassium. Potassium is the key player. So why do you think it would be the key player? Does anyone have any idea?
Smaller in size.
It's the ion that allows for the electrochemical gradient.
Okay. Okay, that's right. So she said it passes easier than Na. So if potassium leaves the cell and Na finds it harder to enter the cell, what would be the membrane potential be? Negative. Okay, that's right. And then also the active transport of sodium and potassium by the sodium-potassium pump will maintain the sodium and potassium concentration gradient. Because if we're moving 3 out and 2 in,
we're not moving it equally. So if they were equal, this would disrupt the cell activity, like muscle contraction, nerve impulses. This would not happen if the concentrations were equal. Okay. Now for cell interactions. So the cells will interact with the environment to maintain homeostasis, which is basically a stable environment. So these interactions can either be direct or indirect. So when we say direct, we're referring to cells directly contacting each other. And when we talk about indirect, that means that they're responding to extracellular chemicals, which are ligands, like neurotransmitters or hormones. Also, we have two main molecule families for interaction. We have the cell adhesion molecules and we have the plasma membrane receptors. Okay, so for cell adhesion molecules, I took these points from your book. Guys, it's very important. I want you guys to note every single point and memorize it by heart.
Take a picture.
Yeah. Okay. So cell adhesion molecules are often referred to as CAMs. They're sticky glycoproteins. They help in anchoring cells to each other or the extracellular matrix. They help migrating cells past one another. They send SOS signals to white blood cells to injury or infection. So you're going to study this in Bio 2, but basically that's wherever there's an injury, CAMs will basically lead the white blood cells to that site of injury. And then lastly, they act as sensors, transmitting extracellular changes into the cell to trigger migration, growth, or specialization. Okay, now to the roles of plasma membrane receptors. It has two roles. First, the plasma membrane receptors are integral proteins, like what Leen said. So they have two functions, either contact signaling or chemical signaling. So when we say contact signaling, that means that the cells will come together, okay? While we say chemical signaling, that means that we have a ligand, this ligand will bind and it will initiate a response. And also, one more thing to note down is that if we have the same ligand, it can bind to different cells and still give different responses. So for example, acetylcholine is a neurotransmitter. When it binds to skeletal muscles, it stimulates it. But when it binds to heart muscles, it inhibits it. Okay, I'm going to go to the picture first and then we'll come back here. Okay, now for G proteins. Right. So first of all, we have G-coupled protein receptors. They're right here. We have ligands, which we said are hormones or neurotransmitters. They're going to bind to this receptor and they will activate it, and this will cause a change in the shape. Okay? Once it does, this will cause the activation of the G protein, and once G protein is activated, it will let go of the GDP and it will bind to GTP, which is a stronger energy source. And as I said, GTP is the same thing as ATP, it's just a different nucleotide, so it's still an energy source. Once that happens, the activated G protein will then activate or inactivate an effector protein, which is here in your book, an enzyme. So this activated enzyme or inactivated will then activate other reactions that produce second messengers. And I want you to know two second messengers, it's very important, the cyclic AMP and calcium. And then these second messengers will then activate other enzymes and the cascade keeps going on. Okay. Now let's start with the cytoplasm. So the cytoplasm is the space between the cell membrane and the nucleus. The nucleus is in the center, and surrounding it you'll find the cytoplasm. And it's the site where most cell activities will happen, and it has three main components. We have the cytosol, which is a jelly-like fluid. It's mostly made of water and solutes. Then we have the organelles, which are mini organs. Each one has its own specific job inside the cell. And then lastly, we have inclusions, which are stored substances. It could be fat granules, it could be glycogen granules, it could be pigments like melanin, and they're not always present in cells. It depends if the cell needs them or not. Okay, so let's start with the first organelle, which is the mitochondria. So I think we're all familiar with the phrase mitochondria is the powerhouse of the cell, right? Okay. So the mitochondria will produce most of the cell's ATP, or energy, and its number can either increase or decrease depending on the activity of the cell. So if we have cells that have high activity, like kidney, liver, and muscle cells, they'll have more mitochondria, while the cells that has lower activity and does not require a lot of energy, they'll have lower, like lymphocytes. And now for the structure, the mitochondria is double membrane. So it has an outer membrane, which is smooth, and then we have the inner membrane, which has folded cristae. And the cristae are just foldings, and they're just there to increase the surface area. And then in the inside, we can find the matrix, which is a gel-like substance that has enzymes, DNA, ribosomes, and so on. One thing to note down is that mitochondria have their own DNA, their own RNA, and their own ribosomes. And then we said we can increase or decrease mitochondria. So how this happens is whenever a cell requires more ATP, we're going to go through a process called fission. So fission is basically that the mitochondria will split in half, and then each half will grow into the former size all over again. Okay, now for ribosomes. So ribosomes are small dark staining granules. They're composed of two components. We have rRNA and proteins, okay? So the ribosomes are the site for protein synthesis. So all proteins are made in the ribosomes. So ribosomes come in two shapes. They can either be free ribosomes, that mean they're floating freely in the cytosol, or they could be membrane-bound ribosomes. Now, if we say free ribosomes, that means that the proteins they're producing will either go to the cytosol, to the mitochondria, or some other organelles. While we say membrane-bound ribosomes, they're attached to the rough endoplasmic reticulum, which we will talk about in a bit. But the proteins that they synthesize are either destined in incorporation in the cell membrane, or they go to lysosomes, or they're just exported out of the cell. Okay, so for the rough endoplasmic reticulum, you need to note down that it's sac-shaped, and why we call it rough is because it's studded with ribosomes all across it. So if it has ribosomes, what do you think the main function of the RERT? Okay. So the proteins that it synthesizes will then be packed in vesicles, and it will then be transported to the Golgi apparatus, which we'll cover in a bit. Also, the rough endoplasmic reticulum can form integral proteins and phospholipids. Okay. Now for the smooth endoplasmic reticulum, it's smooth because it's not studded with anything. There is no ribosomes. And it's shaped as tubules. There's a lot of reactions that the enzymes in the smooth endoplasmic reticulum will catalyze. Let's start with the first one. They will metabolize lipids, synthesize cholesterol and phospholipids. They can synthesize steroid-based hormones. So when you say steroid-based hormones, we're talking about estrogen and progesterone. And then they can work with detoxification. They basically break down drugs. And this mainly happens in the liver and kidney cells. So if you drink a lot of alcohol, this alcohol will affect your liver, right? So you'll find the detoxification highest in the liver. And then lastly, they store calcium ions, and this happens mostly in the muscles. And in muscles, the smooth endoplasmic reticulum has another name. It's called the sarcoplasmic reticulum. And this stored calcium will then lead to trigger contraction. Now, one thing you need to remember very well, that the smooth endoplasmic reticulum has nothing to do with protein synthesis. Okay? طيب فإذا نحن هلا حكينا إنه which ER will synthesize steroid hormones, what would it be? Okay. Proteins. Detoxification. Okay. Now for the Golgi apparatus. So keep in mind these three words. It modifies, concentrates, and packages the proteins. Now we said the rough endoplasmic reticulum will form the proteins, right? And we said it's going to be transported through vesicles to the Golgi apparatus. So these vesicles will move to the Golgi apparatus through the cis phase. Once they're there, they'll have three fates. They can either be out for secretion. They'll be packed in secretory vesicles and secreted out of the cell. They could be destined for the membranes. So this would specifically be for phospholipids. So we said that the rough endoplasmic reticulum can also form phospholipids, right? So these phospholipids will also pass through the Golgi and then out of the Golgi from the transphase to the plasma membrane and form the plasma membrane. And lastly, we said digestive enzymes. The vesicles will bind with the lysosome and the proteins will be digested. Okay. Now for peroxisomes. So they're spherical membrane stacks and they have powerful enzymes. So one of the first enzymes it contains are oxidases. So does anyone know what free radicals are? Okay, that's right. So if we have a lot of free oxygen, known as free radicals, it's very toxic to the body. So we'll have to convert it to hydrogen peroxide, which is H2O2. Okay? This is done by oxidases. But also the accumulation of H2O2 is also harmful. So catalases will then convert H2O2 into H2O, which is water. So the role of peroxisomes is very important. Please note these down. They're very, very important. First, detoxification in the liver and kidney cells. Where did detoxification also happen? Okay. So try to link stuff together. And then energy metabolism, which is breakdown and synthesis of fatty acids. And lastly, they're formed by fission. So where did we also use fission? Okay, so we said fission is the pinching in half and then each one grows into its former size. Okay. So we've been talking about endocytosis, exocytosis, and we've mentioned lysosomes. So what is the function of lysosomes? Okay. So lysosomes will have special enzymes called hydrolytic digestive enzymes. These enzymes will break down whatever enters. Okay? So the functions, they're abundant in phagocytosis. Phagocytosis is when macrophages engulf particles. So that's when lysosomes come in place. They fuse with the phagosome, with the macrophage, and the inside will be digested. And they work best in acidic conditions. So the enzymes in lysosomes are acid hydrolases. Now we have special membrane adaptations. First is the proton pumps. So are protons acidic or basic? Acidic, right? So if we have a proton pump on the membrane of the lysosome, that means more protons are going to enter the lysosome, making the inside of the lysosome more? Okay. And then we have selective permeability. So lysosomes will trap dangerous enzymes. They will digest them, but they won't let them leave until they're fully digested. So these dangerous particles are not going to leave anywhere until they're fully digested. I'm not going to go through this because it's mostly stuff you know. Does anyone know what autophagy, autolysis is? I think you took this in Biochem, chapter 3. Yeah? Yes? Yeah. Okay. This paragraph is very, very, very important, guys. I want you guys to memorize this like your name. كتير مهم, okay? I'm not going to go through it. Read through the parts alone. But it's mostly stuff that you already know from Biochem, okay? So now moving on to the last part, which is the cytoskeleton. The cytoskeleton is formed from three stuff. So let's start off with the microfilaments. So microfilaments are basically strands of protein, and the subunit here is actin. So we have strands with beads on it, and these beads resemble actin subunits, okay? So as we said, they're the thinnest of the cytoskeleton. They're semi-flexible strands, and they're involved in cell motility. So if we say they're semi-flexible, meaning they can change shape. So this will keep the shape of the cell. Now they have several roles. First, cell division, which my friend Reem will go over in her part. Then we have cell movement. They're going to attach to adhesion molecules. And once they do, this will help the cell move throughout the extracellular fluid. And then they're also involved in membrane changes, endocytosis and exocytosis, and also muscle contraction. So we said we have actin, which is the protein. We also have another muscle protein, which is called myosin. So once they interact together, this will generate the contractile forces, which causes contraction of the muscles. And lastly, they're for structure support, so they maintain the shape and distribute tension in the non-living cells. Okay, now moving on to the second one. We have intermediate filaments. So unlike microfilaments, intermediate filaments are very tough. They're not dynamic. They're not flexible at all. They're not flexible at all. So think of them as the cables or steel cables inside the cell. Okay? So intermediate filaments are the most stable and permanent. Please know the keywords, because these keywords will help you or guide you through answering the questions. They're going to attach and anchor desmosomes, which what Leen explained, and she said that desmosomes are there for resisting pulling forces or for mechanical stress. So these intermediate filaments will help anchor the desmosomes. And lastly, you can find them in neurotransmitters and nerve cells and keratin. So one of the proteins that could form these intermediate filaments are keratin. For the last cytoskeleton molecule, we have the microtubules. So the microtubules are the largest in size, and they're very hollow. They have cavities in between. Stuff can pass through them. So think of them as a railway that stuff could be transported through. So they're made of the protein tubulin, and they are very dynamic organelles. So meaning they can change, they can move. They're not rigid like the intermediate filaments. And they're mostly bound to organelles like mitochondria, lysosomes, and secretory vesicles, because these organelles require movement. And we said microtubules will allow organelles to move throughout the cell. يعني أكيد مثلا نحن we have the secretory vesicle, of course it's not moving on its own. يعني there's something holding onto it, right? And then lastly, they have tiny protein machines called motor proteins, like kinases, dyneins, and others. Now we have cilia and flagella. I'm pretty sure you guys know this. So let's start first with cilia. There are cellular extensions. They occur in large numbers on exposed cell surfaces, and their function basically is to propel substances in one direction only. They're not multi-directional. Okay? One of the examples is in the respiratory tract. So in your respiratory tract, you can have dust, you can have mucus. So the cilia will help move this to the outward, so it wouldn't enter your body. And we'll talk about the formation in the next slide. Then we have the flagella, which are similar to cilia, but they're much longer. So we said cilia will move substances, right? But for flagella, it propels the cell itself, not other substances. Okay. Now for the ciliary action. First, let's look at the structure of the cilia. We have 9 duplets of microtubules. So each duplet has 2 microtubules, and then we have 2 centers in the middle. So these microtubules, the duplets, will attach to each other by dyneins, which are flexible motor proteins. So once they do, we're going to have 2 actions. One is the power or propulsive stroke, and the other is the recovery stroke. So power or propulsive stroke is when the cilia is going straight, and then recovery stroke is when it's returning to its bending position. And the dynein is the dynein here that connects the microtubules to each other, is what helps to form this action. So if dynein wasn't there, they would be moving randomly and they wouldn't be bending over. Okay, and lastly for microvilli. Microvilli are just finger-like projections of the plasma membrane. They're also referred to as the brush border of cells. They increase the surface area. They're found in absorptive cells like the intestine, the kidney tubules, and they're formed of bundled actin filaments. So what did we say bundled actin filaments are called? Which type of the cytoskeleton? No, microfilaments. Okay. So the actin filaments, they will extend into a dense network called the terminal web, and then actin here will function as a mechanical stiffener, helping maintain the shape of the cell. Okay. Which of the following pairs of terms is mismatched? If you want to correct it, what would you change it to? Yani A is wrong. We said smooth endoplasmic reticulum does not produce energy. What does it do? Okay, that's right. for transporting the egg. Which of the following cytoskeleton structures is linked to this movement? Okay. And we said how are they arranged? How are the microtubules arranged? Yes, that's it. Okay, now my colleague Reem will carry on. Can you hear me? Can you hear? Can you hear me? Yes. Okay. So now we're diving into the nucleus, and as you guys know, the nucleus is the most vital organ in our cell. So can anyone tell me what are the parts of the nucleus? And no, like what is the main function of the nucleus? Like what does it do?
It holds the DNA and it's the cell's control center.
Yes, right. So the nucleus, it protects the DNA from any damage, and it also regulates gene expression. Now we have three parts that are mostly important in the nucleus. Does anyone know like the three layers of the nucleus or the three main parts of the nucleus? Yes. Okay. So we have three parts. We have the nuclear membrane or the nuclear envelope, the nucleolus, and the chromatin. Now I'm going to start with the nuclear envelope. So the nuclear envelope is a double membrane barrier. It's like the mitochondria, as my friend Tala explained before, and it has a fluid-filled cavity. Now the nuclear envelope is not just a wall. Like think of it as a regulated gate. So first we have two parts of the nuclear membrane. We have an outer membrane and an inner membrane. And the perinuclear space is where the fluid is there. So outer membrane is continuous with the rough endoplasmic reticulum, as you guys can see, and it also has studded ribosomes within it. Now, moving on, we have the inner membrane. The inner membrane is like below the outer membrane, and it is lined with a very special structure. It's called the nuclear lamina. Nuclear lamina is a complex of proteins. It's a network of lamins. The protein is called lamins, which basically regulates what enters and what leaves the cell. Okay? And the nuclear membrane is actually not sealed, fully sealed. It has pores, which are called nuclear pores. And within the nuclear pores, there is a lining of proteins or a complex called the nuclear pore complex, and it actually regulates the movement of molecules. Do you know an example of molecule that can leave and enter the cell? Any example, like the nucleus specifically? Yes, mRNA, proteins, tRNAs, and these molecules should be regulated because as I said, the nucleus has the DNA, which is a very important and vital part of our cell. Okay. Now the nuclear envelope is selectively permeable. That means small molecules can pass and enter with two types of transport. So small molecules can pass through pores easily. Do you know what type of transport is this? Simple diffusion. And we have also proteins and RNA, and these proteins and RNA are very large molecules, so they need a helper, which is transport proteins, and they use energy. What type of transport uses energy, or ATP? Active transport. You have to link so that you can make the connections make the topic more easier. And then the last part here, we have the nucleoplasm, which is a jelly-like fluid, fills the nucleus, and it also contains salts and nutrients. It's just like the cytoplasm of the cell, except it's just specific for this nucleus. Okay, the second structure is the nucleoli. Nucleoli or nucleolus. They are dark and spherical bodies. This is where the ribosomal subunits and the ribosomal RNA, they're synthesized there. And they're actually found like within the nucleus, and it can, like, a nucleus can have one or more nucleolus. Okay? And as I said, it is where the ribosomal subunits are assembled and RNA are made. And also, they live within the nuclear pores, and then they form the ribosomes in the cytoplasm, which will lead to the process of translation, as I'll explain later. Okay, now we also have chromatins. Chromatin are like, basically it's our DNA. It's a protein-gene complex, and they appear as fine threads in the nucleoplasm, and they contain, they have 3 compartments. Like, the chromatin is made up of 30% of DNA, which is our genetic material. It's also made of 60% of histones, which basically is the protein that will basically will package and regulate the DNA. And we also have 10% of RNA, the newly formed or it's being formed. Now the chromatin is like a tent-like structure. We can't leave it in the nucleus like it's floating within the nucleoplasm, because if we want to perform DNA replication or cell division, this would cause the DNA or the chromatin to be broken down, or it can be tangled, which will cause a damage. So they have to be wrapped, and they have to be in like it's a structure, يعني مضغوطين, okay, and like compressed. So we have a certain type of protein which is called histones. They would wrap around the DNA to form nucleosomes. Now nucleosomes, it's just like a bead on a string. So it's like multiple histones together, packed together to form the nucleosome. Now histones, they have groups that will indicate if the cell is active or inactive. So if you found that the histones have methyl groups, that will indicate DNA inactivation, which will form the condensed chromatin, which is an inactive and dark staining. You can see this. Okay, you can see this, the condensed chromatin. It's before the DNA replication, cell division, you would find them there. Okay, and we have acyl groups. It's for DNA exposure and gene activity. That means that they will form the extended chromatin, which is active and invisible under the light microscope. And as I said, we have to package, or we have to put them in a place where we can see them before cell division. So the chromatin will condense and to form chromosomes. Do you know how many chromosomes does the human body have? 46. Okay. So it's, as I said, it prevents forms into chromosomes to prevent tangling and breaking. Okay, we have a question. Which of the following correctly matches a nuclear structure with its function? Who wants to answer? Yes. Yeah.
D. Which of the following correctly matches a nuclear structure with its function? Nuclear pores allows molecules to move between the nucleus and the cell.
Yeah, that's correct. So the answer is D. Okay, another question. Place the following structures in order from least to most complex. Yalla guys, we need someone to participate. Yes? Okay. So we start from least to most complex. You said A, which is the right answer. You would first start. Okay. You would start with your genetic material, the DNA, and then you would condense it to form a chromatin, then a nucleosome, and then a chromosome. Oh, wait. Okay, let's see. Basically, it's DNA, nucleosome, and then the nucleosome will form chromatin, and then the chromatin will form chromosome. You have to know the order because they're gonna get a lot of questions. A lot of questions came specifically from this part, like the order.
I have a question.
Yeah.
What was nucleosome again?
Nucleosome is basically when the DNA is wrapped around 8 histones. This is the nucleosome. And then multiple nucleosomes will condense together to form the chromatin. And the chromatin will also condense to form the chromosome. Okay, now we're moving to the cell cycle. Now the cell cycle is a very important thing. You guys, you have to focus. This is like our cell quiz was mostly from this. Okay, so we have two main periods. We have interphase, which is the growth and the normal activity, which is indicates for growth and normal activities, and mitotic, which is cell division, the actual cell division. So what happens in interphase is we have interphase is only for preparation for for the division, for mitosis. So here basically the cell will just perform its regular activities like growth, like producing proteins, synthesizing enzymes, all of these stuff that would like make the cell metabolically active. Now the interphase, some people might say it's a resting phase. That means like it doesn't, like it's not dividing, but basically it's not a true resting phase because the cell is active but not for cell division. It's preparing itself for cell division. Now we have 3 phases for the interphase. We have gap G1, S, and G2. Now G1, which is also called gap 1, this is where most of the activity like of the cell happens. It's basically where the cell grows. The cell also synthesizes proteins and the organelles like the mitochondria, the plasmic reticulum, whatever. Now the G1 is variable in length. So in some cells it might be like 10 minutes, or other cells it might take years. So like it's variable. But if you see a cell that stops dividing, like it's not metabolically active, it doesn't form organelles, it doesn't like just this, it's the normal activities, it will enter the G0 phase. So the cell is actually like not active at all. Okay? And then we have the S, which is the synthesis phase. It's basically DNA replication, and like what's DNA replication? Do you know like an example of like the general idea of DNA replication? Yes, so it's basically just making an identical like DNA for like 46 chromosomes. We will also be doubled to 46 chromosomes, and then we're just making copies of the same DNA. And then we have the G2, which is the gap 2, and this is the final step before entering the mitosis. Okay, so here the cell should have their proteins and their enzymes synthesized and completely like active. The centriole replication is completed. I'll talk about this later, but basically centriole is important for mitosis as it will produce the mitotic spindle. And then we have the G2 and checkpoint. This checkpoint is very important because, for example, if we have the cell and like most of its organelles are not fully developed, it cannot go to mitosis. It cannot be ready for the cell division, because if we divide the cell, the other cell will one of the cell will be okay, completely fine, but the other cell will be defective. So we don't want any errors in the division process. Okay, we have a question. During which phase of the cell cycle there is no proof of cell division activity?
G0.
G0. Okay, yeah. What happens in G1?
G1 growing. Cell
Growing.
doing.
What about S?
DNA
DNA replication. What about G2?
Getting.
Yes. Okay, now we have DNA replication. So DNA replication is the process where you guys, please focus. Okay, so DNA replication is where the cell starts to duplicate its genetic information, and this would have like, it will form an identical copy. Later, when we want to divide the cell into 2 cells, it would have the same identical genetic material. Now, DNA replication, as we said, it occurs in S phase, so it's part of interphase. It's still not the actual cell division. Okay, and also starts with multiple origins. Why does it start with multiple origins? Faster replication. Basically, our human DNA is very long. So if we start from one origin, it would take us years, maybe like a lot of years, to actually like duplicated. So we need multiple origins to fasten the process. Now let's start with the actual steps of replication. First step is the uncoiling. So we have the DNA, which is a double helical structure, right? So it is coiled and it's like going as a helix. We need to uncoil, we need to uncoil the DNA to make them as single strands. So the uncoiling enzymes will unwind the DNA and it will form a replication bubble. Everything that happens during replication is within the replication bubble. Okay? Now, after we uncoil the strands and they became single-stranded, we need something to separate the strands. Because as we know, between the bases we have hydrogen bonds. Okay? And these hydrogen bonds should be broken down so that the strands would be separated. So the hydrogen bonds break and the strands unzip at the replication fork. This would form the replication fork where we will have the promoter region, and this is where the DNA replication will start. Okay? Now the third step is the assembly. Assembly is basically after uncoiling and separating, we have now the DNA strands are ready to start replicating. But we need an enzyme. This enzyme is called DNA polymerase. This enzyme will it will start synthesizing new strands, complementary strands, okay, and they will attach them to the template strand. So I didn't mention this, but when we separate the DNA strands, we would take the template DNA, like the one that we use to make the new DNA strand is called the template. The original strand is called the template strand. And the new formed one is called the complementary strand. Okay? So DNA polymerase will build new complementary strands using all as templates. Now, do you know in what direction, like how many directions that the DNA polymerase moves? 3 to 5. Is it one direction or 2 directions?
No, it's one direction of duplication, but it has 2 directions, so one is lagging, one is leading.
Yes. So it moves in one direction. That will lead us to form 2 strands, the leading strand and the lagging strand. So the leading strand is synthesized continuously. That means it moves within the same direction as the DNA polymerase, and the lagging is synthesized in fragments. Because it's moving to the opposite direction, it's not continuous, so it has to stop at some point and start duplicating at another point. And these fragments are called Okazaki fragments. Okay? Now this process of replication is semi-conservative. Semi-conservative means that it uses an old strand, which is the parental strand, and it will use one new strand. So the new DNA strand is called a semi-conservative strand also, because it has one from the old, from the parental side, and one from our DNA polymerase. Now the last step is the restoration. Basically, after forming the nucleotides, after forming the new strands, we need to join them together again by an enzyme. Okay, DNA ligase, they join the fragments and reforming the double helix. So we have to also again recoil the DNA, and this enzyme is responsible for this. Okay, this is a picture. This is a summary of what I just explained. You have to it summarizes everything I said, the complementary, the leading, the lagging, the replication bubble. But please go back to your book and look at this, because this will give you a good image. Like while you're in the exam, you can still remember the image. Okay. Since the two, can anyone read? Okay.
Since the 2 strands of DNA retain two products formed as the result of DNA replication, it is called semi-conservative.
Yes, right. Okay. So we're starting with cell division. So please focus, because this is, I can't emphasize how important this part, specifically the later slide you will see, I used the same figure from your book, because most questions of mitosis came from that figure. So you have to know everything in detail. Okay? So mitosis, go to the question. I have a question before mitosis, which of the following best describes the process of mitosis? 2 daughter cells with 2 sets of chromosomes are produced. Yes, correct. Okay. Now we're starting with mitosis. The first part is interphase. Now interphase is not a part of mitosis or cell division. This is the phase where it prepares for cell division. So as I explained before, interphase has 3 phases. It's just where the cell grows, the cell develops, synthesizes proteins, enzymes, and it's just ready to get divided. Okay, now we're moving on to the next phase. Okay, now we have prophase. Now prophase is divided into 2 periods, early and late. And prophase is actually the first step of mitosis. Okay, so what happens here, I'll explain from the diagram here. What happens here, first, your chromatin will condense and coil to form chromosomes. They are darkly stained so that you can it can easily get divided. And we also have that each chromosome has 2 chromatids. Okay, so a chromosome, it's like an X shape, and then you have 2 chromatids joined by a centromere at the middle of it. Okay, and then as the chromosomes appear, the nuclei will disappear, and the centrosomes. Okay, now let's go to the centrosomes. Now the centrosomes, these are the centrosomes. Okay, these are the focal points where, like, basically the cell would start forming microtubule assembly, which is called the mitotic spindle, where basically this will help in the division of the nucleus. Like the nucleus would be separated by these structures. And the microtubule arrays are called asters. We have something that's going to come out like small extensions from the centrosome. Okay, these are called asters. You can think of them as like stars because it's like almost similar. So it's like a star, almost like a star. It's like a star. It's basically extensions coming out from the centrosome or the microtubule assembly. Okay, now we have the late prophase. This is where the envelope breaks up because we want the mitotic spindle or the microtubule arrays. We want them to like go through these chromosomes and start the division, the actual division. So the envelope will break, and then some of the growing spindle microtubules will attach to kinetochores. Now here, the spindle pole will synthesize 2 types of microtubules. This first type is called the kinetochore, which will start like it will come from the mitotic assembly, and then it will go through the centromere that is in the center of the chromosome, and then this will attach to a protein in the centromere, which is called the kinetochore. This is a protein. And we have the other type, which is called the non-kinetochore microtubules, which basically slide past each other, and these would make the cell divided into to the poles. So like they would go away. Like imagine that this is the mitotic spindle, and then this would just move away from the chromosome. The non-kinetochore one, the one that are not attached to the chromosome, they would move away, and they would like separate to basically make 2 poles of the cell. Because later we will see that the cell will be divided in half, and then each cell will have 1 microtubule or something. Okay. Now we have the metaphase. So as I said, the mitotic spindle will produce 2 types, the kinetochore and non-kinetochore. Now both of them, they will help align the chromosomes into the equator of the cell, and this would form the metaphase plate. It's an imaginary axis. It's not there, but it will be like in the middle of the cell, and the non-kinetochore microtubules would also lengthen more. So they would basically, what happens in metaphase, the chromosomes are not in the middle. That's it. Like this is the important. Okay. Now we have anaphase, which is the shortest phase of mitosis, and basically what happens is this is where the chromosome will separate, actual separation of chromosomes, and it will be separated, and it will go like through the opposite poles. So what drives this separation is the centromere. Centromere will lead the way to the microtubule. The kinetochore, like تصوروا انتم مثلا kinetochores, they want to go back to their original place. Like خلاص, I want to go back. So they are connected to the centromere. So the centromere are like, okay, let's go back. So they're gonna go back to the microtubule, and the other ones, the non-kinetochore ones, they would lengthen more to separate the cell more. Like the cytoplasm will be much more separated. Like it will be very bad, يعني عن بعض. Now we have the last step, which is the telophase. It's like the reverse of prophase. Basically, what happens, the organelles would reform, the nuclear envelope will also reform, and also the nuclei will reappear. And you will see that we have at each, like when it's separated here, you'll see we have one microtubule assembly at each cell. So that's why it gets separated, or it gets like very far away from each other. Okay, and then here mitosis is ended. خلاص. Telophase is the final phase. So mitosis is ended, but you can see that it's still not completely like separated. Why? Because your cytoplasm is still connected. It's just that you have the nuclei are separated from each other. So this is a binucleate now. It's not a uninucleate. So what we do is we have another process called cytokinesis, which is the division of cytoplasm. We will have the microfilaments, the actin microfilaments, and they will form a ring, which is called cleavage furrow. And this would actually indicate the division of the cell. Like this is the cell, cytoplasm of the cell is divided. Okay. Which of the following is correct? The order of the phases of cell division. Okay. I want you to mention, like, to tell me what happens in each step. Yalla, since the answer is there. Okay.
Fema, A.
Okay, Fema. What happens in interphase?
You prepare for the mitosis.
Okay. What about?
The spindle fibers appear.
Yes. Metaphase.
Metaphase, it lines it up in the middle.
Yeah. Anaphase.
Anaphase, you start pulling. بعدين telophase, nucleus or DNA is separated.
Yeah, okay, right. So this is a way to remember it, PMAT, okay? It's easy to remember, so I use this in the exam. Okay, moving on. Okay, now we have the control of cell division. So the cell division, they have internal and external factors. And this will tell the cell if it will, like, okay, start dividing or no, you cannot divide, you have to stop. So first we have cell size. As you guys know, the cell will start to grow and interphase the growth of the cell and everything. So when the cell grows, actually the volume of the cell will increase faster than the surface area. And so if the cell is actually very big and large, you know that the cell gets its nutrition through the surface area, through like diffusion. Okay? So if the surface area is actually large, then the cell will not get its nutrition faster and it will die. Okay? So the ratio of the cell surface area to the cell volume affects the cell size. Now we have chemical signals. Now cells receive signals from growth factors and hormones, and these will indicate if the cell should start dividing and growing. And we have space availability. So when the cell will stop dividing if the cells are touching each other. Okay? And this is called contact inhibition. This is a phenomena that will tell the cell you have to stop because there is no space for you to divide more, and this would cause damage to the other cells. Okay. Now we have checkpoints. We have two checkpoints, G1 and the G2M. Now G1 is called the restriction point. This is where it's called a restriction point. This is where if the cell is actually has the correct amount of organelles, if it grows group well, and if we have the adequate amounts of energy reservoirs, okay? And then G2M it's where checkpoint. We said this before. It's where the cell it will determine if the cell is correctly replicated, if we have these enzymes and proteins. And then we have two proteins, or two types of groups of proteins, cyclins and CDKs, and cyclin destruction. Now cyclins and CDKs. CDK is called the cyclin-dependent kinase, and it's activated by cyclin. So if the CDK is bound to a cyclin, it will initiate a reaction and like a signal transduction. You know what signal transduction is, yeah? So it will tell the cell that okay, you have to start dividing. And we have cyclin destruction. After mitosis is done, these enzymes will destroy these cyclins, and the mitosis will end. Okay. So a gene, this is like before we start the next part. A gene can be best defined as B. A segment of DNA that carries the instructions from one polypeptide chain. Yes. Okay. Now we're moving on to messenger RNA, as it carries instructions from DNA to building protein. So now we want to, after the DNA is replicated and everything, we need to start forming proteins, because proteins are the most the important molecules for any cell interaction, any cell activity. Okay. So as you guys know, we have our human bodies have a lot of thousands and thousands of gene coding protein encoding genes, and then the mRNA it uses a genetic code A, G, T, the gene itself uses A, G, T, and C. These are bases, and each 3 bases, they are called a triplet. Okay, and this would code for one significant amino acid. So each 3 bases would code for 1. So the ratio is 3:1. 3 bases, one specific amino acid. Now the gene consists of coding regions, which are called exons and introns. I will explain them in transcription now. And introns can regulate gene expression, enable different proteins from one gene, and promote evolution. So here I don't want you to go into introns because they're very detailed and they're very complex structures. But basically introns, why are they there? We remove them, but why are they there? It's to regulate the gene. When we transcribe the gene, it will not encode this اشياء غلط يعني اوكي فشو اسمه فمشان يكون the gene it's specific and regulated. Okay. Now we have the role of RNA in protein synthesis. Now RNA, it acts a decoder messenger for DNA. It basically takes the information from DNA, it will decode it, and it will give this information to the ribosomes. So RNA differs from DNA. Do you know what are the differences?
Yes. Yes.
I want new hands. New hands.
What's the question?
What's the difference between RNA and DNA? Please guys, this is also in biology.
Double strand, single strand.
Okay. What else?
Sugar type.
Sugar type. What does RNA have? Okay, and the bases. What does it have? Different from DNA?
DNA has thymine.
Yeah.
RNA has uracil.
Yes, good job. Okay. Now we have 3 types of RNA. We have mRNA, rRNA, tRNA. Now mRNA, it carries the coded information to the cytoplasm. This is where our protein synthesis happens. And we have RNA, which forms the ribosomal subunits, and tRNA, which carries amino acids to the cytoplasm. Okay. Now all RNA types are formed on DNA in the nucleus. Where does translation happen? Cytoplasm. Okay. Now polypeptide synthesis is involved with 2 processes. We have transcription, which is DNA's information is encoded in mRNA. So the mRNA will take the information from the DNA and it will leave the nucleus and it will go to the cytoplasm where translation happens, which basically mRNA information is decoded to protein synthesis. They will take this information and they will start synthesizing polypeptides. Yeah. Okay, question. Which of the following structure carries a sequence of 3 nucleic acids called a codon? Yes. Yes, messenger RNA. What does transfer RNA carry?
Anticodon.
Anticodon. Okay. Okay, now we'll start with transcription. I'll explain from the figure in your book, because all the figures are important. They're very important, specifically the last part. Okay? Okay, now we'll start with transcription. So first, we have our DNA, and there's an enzyme, a new enzyme. It's called RNA polymerase. This would come, and it will bind to a region called the promoter region. And the promoter region, it will tells the RNA polymerase, yalla, let's start transcribing, and let's start to form polypeptides, okay? So mRNA synthesis is at the start within the promoter region, and we will use the template strand again, okay? The template is the original strand, so it will use it again to start encoding. Now we have the second step, which is elongation. And basically elongation, it's where the RNA polymerase moves along the strand, and then it joins like RNA molecules together to form the RNA complementary to the DNA. Okay? And this, it elongates the mRNA transcript one nucleotide at a time, and unwinding the DNA double helix in front and rewinding it behind. Okay. هشرحها مثل قصة. So احنا خلاص we started, okay, we separated the two DNA strands, and then we're going to use the DNA, the template strand, and the RNA polymerase would be bound to it. Now we said the promoter region is where the transcription actually starts, and now elongation is RNA polymerase will produce the complementary strand of RNA that's complementary to the DNA. Okay, and then after that, termination, which is the last step in transcription. This would tell the RNA polymerase, خلاص, you have to stop transcribing. We have our mRNA. So there is a signal or a sequence of nucleotides, and it's called the termination signal. So what starts transcription? Which region? Promoter. What terminates the transcription? Termination signal. Okay. So guys, this is very important. I wrote this in foundation. This was with Dr. Aboudraz, and basically, you have to know which one is identical to which and which one is complementary to. So, we have template is the original strand, okay, and we have the mRNA. So, template and mRNA are complementary. That means like it binds to it, and like it's different bases, but it's complementary to it. Like when we have A, it will give us what? The mRNA. U. Okay. Now we have the template, the mRNA, and the coding. Now the coding, in the DNA, we had two strands. As I said, it's a semi-conservative. So one is template and one is new. The new one, I said it's coded as a coding strand. Okay. So the mRNA and the coding are identical. They have the same basis, the same alignment. Okay. And then we have the template and coding are complementary. Coding is another, like it's the same, as I said, mRNA and coding are the same, so it obviously will be complementary to it. And then we have the tRNA, I'll explain later. tRNA and template are identical. They have the same alignment. So you have to know this, like you have to. Take a picture if you want, because it's really important.
We're gonna send you the slides later.
Yeah. Today. Okay. Which of the following statements is related to transcription? Can anyone read the choices? Okay.
Which of the following statements is related to transcription? RNA leaves nucleus as a free mRNA. DNA polymerase makes the complementary RNA from DNA. It requires a promoter and a terminator. Introns have the code for protein sequencing.
Yes, it's C. What does DNA polymerase do? If you want to correct it. Uncoiled. And does it make RNA? Which enzyme is used to make the complementary RNA?
RNA.
Yes. Okay. Okay, now we're moving after we're done with transcription, we have to move to the cytoplasm to start translation. But before we start translation, you have to know that the mRNA, it's not functional. Like before it leaves the cytoplasm, it's still not functional. There has to be some editing and further processing of this mRNA, because when we transcribe, like DNA is sequentially transcribed. So we would still have some like parts where we have the introns. And as I said before, introns are going to be removed because they are regulators, but we have to remove them. They're not the code expression, like they don't express the DNA. Okay? So there's an enzyme called spliceosome. This enzyme would snip out the intron parts, and they would splice the exon fragments and join them together to make the functional mRNA. And then the functional mRNA, after that, will leave the nucleus from the nuclear core, and it will reach to the cytoplasm where translation happens. Okay? So translation, it also has 3 phases: initiation, elongation, and termination. Now, initiation, there are multiple steps of initiation. They have to be all met together so that we can start with elongation. So what happens first, that the mRNA will go to the cytoplasm, and then the small ribosomal subunits, we have 2 subunits of ribosomes. It is small and large subunits. So first, the small ribosomal subunit will come and bind to the mRNA, okay? And then the initiator tRNA, which carries the amino acid, will bind to the start codon. Do you know what is the start codon? AUG. So it will come and bind to the start codon. And what amino acid does it contain? Like what amino acid does it carry? The tRNA? Methionine. Right. So it will carry the methionine, and it will bind it to the start codon, and then after that, the large ribosomal subunit will come and it will join. They will be joined together, and then now the ribosome is functional. So now after that, we have to start elongation. So to wrap everything for the initiation, first, mRNA will go to the cytoplasm. Then the small ribosomal subunit will come and bind to the mRNA, and then the tRNA, the initiator tRNA, will bind to the AUG, which is the start codon, and then the large ribosomal subunit will come and join and form the functional ribosome where elongation starts. Okay, next. Now we have the next step, which is elongation, which is where we build or where we synthesize the proteins, like actual synthesis. So it has also 3 parts, three parts. We start with codon recognition. So after the initiated tRNA comes and binds to the P sites, okay? We have 3 sites in the ribosome, A, P, and E. So initiated tRNA starts at the P site, okay? When it will come and it will bind to methionine, then a new tRNA will come, which is called amino acid transfer RNA, which carries the amino acid from its name. It's amino acid, okay? It will come and it will bind to the A site. It will come to the A site and it will bind to the corresponding codon. And then this is what happens first, codon recognition. So it will come to the A site, will bind to the codon, and then we'll start with peptide bond formation. So after the codon recognition, we'll start with the peptide bond. After that, here, you'll see that the amino acid in the P site, where the initiator tRNA was, it will translocate to the A site. Okay, it will go to the A site and it will form peptide bonds by an enzyme called peptidase transferase. This is the enzyme that will synthesize the peptide bonds, and it will join the 2 amino acids, like the amino acids together. Okay, and then after that, translocation will happen. So after it binds to the A site and the amino acid is transferred to the A site, now the existing tRNA, the first tRNA, which had methionine, will exit. It will go to the E, which is exit, and it will just leave. خلاص راح. And then the tRNA in the A site that was in the A site, it will go to the P site. So it's like a loop. It's continuous. They would go from a site to a site. Okay. So it starts with P, then the new amino acid will come in the A, and then the previous tRNA will leave from the E, and then the amino acid that was in the A will go to the P site. Okay? And then we have the last step of transcription is termination, where the stop codons. We have 3 stop codons, and if the mRNA codes for them, there is no amino acid that can be complementary to it. It cannot there is no specific amino acid for this stop codon. So we will have a release factor which will deassemble the ribosomes, and it will release the polypeptide chain along the tRNA. So everything will be released. The mRNA will be released. It can be destroyed or used, because mRNA can be used multiple times, and the new polypeptide chain will form, and it will just go and maintain, do whatever function they are specific for. Okay? Now, which of the following steps is not followed by the elongation of amino acid chain?
Your hands, guys.
Okay. Which is the following? Okay, yes. Copying of genetic code by mRNA. Where does this happen? Yes. Okay. Now, this is important. You have important codons to memorize. Memorize them by your heart. Start codon, which is AUG, which codes for methionine. And then we have 3 stop codons. UAA, UAG, and UGA. You have to know them because they're going to ask questions in the exam about them. Okay, move. Now we have other rows of DNA. So actually the DNA also synthesizes RNAs. Yeah.
I think these are cancelled.
These are cancelled for you?
No, they're not.
You just need to know that they regulate gene expression. You don't need to know what are they used for.
Rough ER is cancelled.
They are cancelled.
Ultra cancelled is cancelled.
Doctor said today they
She
They're
said today.
cancelled? خلاص, we're done.
Guys,